Hi everyone, this is the video project for the UC Davis course. This group is composed of me, Adriana dos Santos Lopes, for the Bioscience Institute at University of São Paulo, Jean Carlos Costa Barros, for the Department of Genetics at Exalc University of São Paulo, Janessa Drestemps for the Department of Plant Biology at University of California, Davis, and Renato Morales Acunha for the Universidad São Francisco de Quito. For the project, we select 3 species: Datura wrighti. Datura chose as a plant of medicinal value for the United States. Anacardium occidentalis. The plant chose as a plant for the food for the Brazil, and Scalesia pendiculata. This plant is chose as an ecological value for the Ecuador.
Hi everyone, my name is Janessa. I'm a first year PhD student studying plant biology at the University of California, Davis, and I will be talking about our US species, Datura wrighti. So Datura wrighti is mainly distributed in the southwestern US, particularly in Southern California, Arizona, and in parts of the deserts in New Mexico. And so it has kind of an interesting ethnobotanical usage by Native Americans in their some ceremonies and mythology. This is because of its interesting hallucinogenic properties that are conferred by the alkaloid composition. Um, and then something that I thought was interesting was this cave painting picture that I found where it has these spiky structures kind of in the bottom left, and it is thought that those spiky structures are a datura fruit. And so alkaloids have been kind of known to have interesting and useful medicinal properties. So for example, atropine is used during surgery to reduce saliva and fluid in the respiratory tract and can also be used to slow heartbeat in an emergency. And then scopolamine is used to prevent nausea and vomiting after anesthesia, as well as narcotic pain medicines. So, although it is known that alkaloids such as atropine and scopolamine have important usages in medicine, there is a large number of uncharacterized or undiscovered alkaloids that have the potential to serve as new medicine, which is where Datura wrighti comes in. And so discovering these new alkaloid structures would also allow for the easier creation of synthetic alternatives to these medicines. And so our research proposal is to use the published Datura wrighti genome on NCBI to find candidate genes based on homology of known alkaloid biosynthetic genes. And then once we have identified those genes, to clone them, and then use a heterologous expression system either in tobacco or E. coli in order to kind of help in elucidating the alkaloid biosynthetic pathway, and then run those products on either the LCMS or GCMS, or if we find a novel product, to do NMR on the bottom right. And so in parallel, while elucidating the biosynthetic pathway, that's really interesting, but a crucial part of this is also looking for bioactivity. And so when novel products are found, we would also conduct bioactivity studies in parallel using purified metabolite. And then eventually, if those have positive results to do in vitro and in vivo studies with mice. So for our research challenges, some technical challenges that I could see being an issue are the lack of genetic information about related species. So Datura stramonium and Datura metel are really the only other species that have published information about alkaloid biosynthesis. And then in addition to that, alkaloids are in general difficult to isolate and tend to be unstable. And so some other challenges that this project may face are that we're working with a non-model species and the project is very exploratory. And so there's not very much preliminary information. And because of that, there's no guarantee that we will find a new medicinal product. And then that could contribute to maybe not getting funded. And so the last issue I could think of was this kind of pervasive underrepresentation of native plants in research, and that contributing to the lack of information about Datura. However, one solution that I did think of is perhaps to collaborate with native tribes in the southwest region of the US and see if they have any resources on Datura.
Hello, my name is Jean, and I will go to talk about one of the species we selected for our project. The species is Anacardium occidentale, popularly known as cashew. This plant is found in Brazil and is native to our country, but not endemic. It is mainly present on the northwest coast in the states of Piauí and Maranhão. Products are obtained from the cashew tree and can be used for various purposes such as cosmetics, pharmaceuticals, and food. Here we have some examples of use in cosmetics or food products. A considerable partition of what is produced is lost. Around 30% of the cashew harvest is lost during transporting or processing. This loss is related to high perishability of this fruit. In a recent study, the authors performed the transcriptome of this species during the fruit development phases. This way, they identify genes related to a maturation phases. Based on these studies, we thought about proposing a complementary study, where we will identify genes after maturation, specifically of days 50, 55, 60, and 65. To do this, we will collect RIP cashews, which will be kept in a controlled and a clean environment. We will the collected cashew apples on the maintenance days. We will perform RNA extraction and the sequencing. With a sequence data, we will use the recently published Cajueiro genome and the tools identify genes differently expressed in the late stage of maturation. The genes will be evaluated through a RT-PCR, and the tools will identify possible candidates for biotechnology tests in the late ripening process of the fruit. Initially, we will have two difficulties during the project, which would be the existence of only a draft of the cashew tree genome and the difficulty of isolation the effects of some microorganisms present in the fruit during the post-harvest phase. So, this way, we can proceed to our next species.
Escaricia pedunculata is an endemic species from the Galapagos Island. Its distribution occurs in humid areas with greater rainfall on the islands of Santa Cruz, Santiago, Floriana, and San Cristobal, at altitudes between 4,000 and 7,000 meters. According to the International Union of Conservation of Nature, Escalesia pedunculata tree is in danger. It is important to say that this species faces a dry season and a wet season every year in Galapagos Island. Escalesia pedunculata have an ecological role as it works as an habitat for endemic species of reptiles, birds, or insects. As we said, these trees face seasonal changes, and it's important to understand how these trees interact with the environment and with their microbiome, who can be playing an important role in a plant resistance through seasonal changes. That is why we propose an analysis of the microbiome of Escalation pedunculata during wet season and the dry season. This procedure will begin with a sampling of the roots in the dry and wet seasons. Once the samples are obtained, the DNA is extracted and we amplify the ITS regions, which allow us to understand the diversity in fungi. And also we amplify the 16S region, which allow us to characterize the diversity in bacteria. Finally, we could analyze the biodiversity of the microbiome with bioinformatic tools. The difficulties we face are the difficulty of obtaining samples from Galapagos due to government impediments and the little information available on microbiomes in Galapagos and on Escalation in general. As a future approach, we can analyze microbiomes of different species of the Scaliaceae genus that inhabit Galapagos.
Thank you all for your attention.